All right, here we go. Getting right to it. So this week we are talking about bacterial cell structure. Bacterial cell structure. Bacteria, we're going to go through normal cells like human cells. We're going to go through bacterial cells, and I'll briefly mention viruses, just so that way we have a clear distinction. But bacteria themselves are very unique cells in their structure and their overall function, what they do in the world. Cells in general, and this is why it was important that we went through the food macromolecules lecture last week. Cells are made up of macromolecules. They're made up of proteins, carbohydrates, and fats. Now, the proteins are going to be obviously different types of amino acids. Now, where are these in the cell? Our tissues throughout our body are going to primarily be protein based. These are all of the major structures, the structural components are going to be protein based. So things like flagella, pili, cell walls. Flagella are like tails, little kind of extensions on the cell that allow it to interact with the environment. Cytoplasmic membranes, ribosomes, and cytoplasm. So all of these general structures are going to be protein based. Cytoplasm is actually going to be the space inside the cell. So like cyto means cell, plasm means space. The cytosol is going to be the fluid that's within there, but the space inside that intracellular fluid is going to have water and protein. Then we have polysaccharides, and these are, of course, multi-chain carbohydrates. They're going to be different types of sugars, and these form capsules, inclusions. Inclusions are like the fat storage of the cell, and then cell walls. Cell walls are only going to be found in plant cells as well as bacterial. They're not in mammalian cells like us. Then we have phospholipids. Phospholipids are going to be the different lipids, and they are fatty acids. Those are going to be primarily found in membranes, and they make up a large component of the membranes to kind of keep it separate, to be able to separate the inside of the cell and the outside of the cell. There's also nucleic acids. Nucleic acids are going to be found in all cells, and these are going to be in the forms of DNA and RNA. Nucleic acids are formed of nucleotides, and what these are is effectively the language and the programming of the cell. Now, cells serve a general function. First, cells synthesize different types of proteins, okay? They're going to be able to intake proteins, synthesize them, and make new ones. By making new ones, they're essentially making different forms of DNA and growing. These proteins are used to make different cell components and enzymes in the body. Enzymes are very important because enzymes speed up chemical reactions in the organism, like digestion. They make reactions easier to happen, and they lower the energy requirement. Otherwise, everything would take super long. Enzymes make things happen faster. They're a catalyst, if you will, like lighter fluid when starting a fire. That's what an enzyme can do for a chemical reaction, is speed it up drastically. And finally, one of the other major cell functions is that they're constantly repairing and replacing cells, so that way they can grow and make sure to replace some of the dead cells or cells that wear out over time. Now, we're going to compare a mammalian cell that you can see here on the left with a bacterial cell. And these have very specific names: eukaryotes and prokaryotes. A eukaryote cell is actually going to be what we are made of. These are animal cells. Plant cells also have this, but plant cells have a slightly different structure, protists and fungi. So these are going to be a lot more typically known living organisms. They are unicellular. They can also be multicellular, and they reproduce both asexually and sexually. The cells have nuclei and membrane-bound organelles. That's probably one of the big key differences, is the membrane-bound organelles and the nuclei. The nuclei itself is going to be composed of different chromosomes. All of the DNA material will be inside of the nuclei. And organelles are like mini organs. Like we have like a liver, we have a stomach, we have kidneys. Those are our organs. Inside the cell, a eukaryote cell, we have things like the Golgi apparatus, the mitochondria, ribosomes. These are going to be different types of organelles, the organs of the cell, and serve different functions. Contrast that with a prokaryote cell. Prokaryotes are bacteria and archaea. These are going to be the prokaryotic cells, and they're single-celled organisms themselves. They don't evolve into other things. They just stay as bacteria, and they reproduce asexually by themselves, just reproducing over time. Now, one of the things that I talked about with the difference between eukaryote cells is these things lack nuclei and membrane-bound organelles. They're primarily just fluid in there. They do have ribosomes, but ribosomes aren't membrane-bound. They have ribosomes, and then they have chromosome or DNA material inside that acts as a programming, but it doesn't really have organelles otherwise. Now, this is where I do want to mention viruses. Viruses have obviously been talked about a lot in more recent time, but viruses themselves are neither prokaryote nor eukaryote. They have genetic material, and that genetic material is actually going to be in this structure that you can see right here, where we actually have this kind of like polygonal structure overall, or this different geometric structure. It's the head or the capsule. This is going to contain the DNA. And there's different structures of viruses. We can see an example of one up here, but this is going to be the actual like diagram of how it would be, as far as like the different structure in this virus right here is going to have on the top of each of these would be little bundles of the head or capsule. And then it's going to be a tail that's going to have different tail fibers and end plates. And these allow it to bind directly to the DNA structure. So viruses have genetic material, and they require a host to reproduce. So if there's no host, if they're just out in the world with nothing living, a virus can't do anything. It needs to have a living host bind to the DNA, and then it can actually start to reproduce and have its overall effect. So it requires a host to reproduce. It lacks cell structure, and it actually lacks metabolism. It is, by its very nature, a virus. It binds directly to DNA, alters DNA to serve its function to reproduce. Once you remove the virus from the host, the virus dies off. Like, it can't do anything. It's neutral. Now, that's the most I'm going to go into viruses. I just want to touch upon them before we move back into bacteria, so you understand the difference. Now, there is a general bacterial morphology of the prokaryote cells. There are foci, bacilli, and different other ones. Now, the foci are going to be like your overall kind of spherical structure of bacteria that can link up together into, different shapes. The bacilli are going to be, I always love this diagram because it's like funny little hot dogs, like a hot dog with a tail. These are going to be elongated ones. These are kind of the more traditional bacteria that you would see, and they can link together. They can have those little flagella tails to allow them to move along. And then in other cases, we have the spirochae, the vibrio, and the spirella. These are just different shapes of bacteria morphology. So each bacteria is going to have a slightly different shape, and that shape is going to dictate a lot of its function. Now, how do bacteria move around? Bacteria move around. They don't just kind of float in your body. They actually have little kind of like tails to propel them. They primarily have they can have a flagellum, or the plural flagella. Flagellum are actually found on a cell inside males' sperm. It's the little tail that actually extends off the main sperm body. And bacteria have this same structure as a flagellum. This is a single tail-like structure. Then we have examples of the flagella. Here we have, you can see a cluster of those tails kind of working together, so where it acts like a propeller on a boat. There's also going to be all of this kind of like hair-like structure. This is the fimbriae that's going to extend off. It helps it work like Velcro to where it sticks onto things. And finally, there's the pili. The pili are going to be longer, thicker forms of fimbriae that are going to be slightly hook-shaped. So it allows it to actually anchor on. The fimbriae will let it stick, and the pili will let it anchor on further from there, so it can bind to the cell structure and begin its reproductive process. One of the other major differences with bacterial cells is going to be and how its membrane is structured. Well, not the membrane itself, but that there's extra on top of the membrane. So In a eukaryote cell, we have this. This is a really great diagram of the phospholipid bilayer. Phospholipid bilayer is a key thing that's in all cell membranes. It is going to be a phosphate. It's going to be a phosphate head with a lipid tail. And we can see that here. We have the hydrophilic head. That means water-loving, philic, hydrophobic or water-fearing tail. So the lipids are going to be here on the tail and the phosphate is on the head. So what it's designed to do, what this membrane's purpose is to separate the outside environment from the inside environment. It keeps everything self-contained. We can see this is a simple diagram of the phospholipid bilayer and a single molecule of it. And then we can see it combined together in a eukaryote cell where we actually have all the different glycolipids, different membrane proteins, et cetera, that are going to be throughout the cell membrane. Now, this is just an example of a phospholipid bilayer. And this is important. We'll expand upon this. So remember that the cell membrane is going to surround cytoplasm of the cell. The cytoplasm itself is actually going to be the space inside. It's a gel-like substance where the organelles lie within the cell. The nucleus itself is going to be in from there, and the nucleus is going to hold the DNA or genetic material. This is like the brain or the communicating center of the cell. And it has its own membrane called the nuclear envelope. And this is made of phospholipids as well. So these phospholipid bilayers are going to make up every single one of the membranes around the organelles. Now, the other organelles, some of them you're going to remember. What is the function of the mitochondria? It's the powerhouse of the cell. Correct. This is where ATP is generated. It is the powerhouse of the cell. There's the rough endoplasmic reticulum. This is going to hold protein-producing ribosomes. The smooth endoplasmic reticulum, do we have a nice picture of it? Membrane, no, not on this one. But there is also a smooth endoplasmic reticulum. It just doesn't have ribosomes on it. That's going to be used to actually create different packages for carbohydrates and lipids in the cell. You have the different ribosomes that are in there. These are going to assemble amino acids into protein chains. Think of these as like little protein coders. They're going to start producing a lot of these proteins. You have the Golgi apparatus. This is going to package the proteins that are produced as far as sealing them off, giving them their own membrane. And then you have lysosomes and peroxiomes. These are going to be produced as well from the Golgi apparatus, and these are going to be the recycle centers of the cell. They contain different digestive enzymes. The peroxiome is actually going to be the hazardous waste bin of the cell. So anything that's really damaging the peroxiome is going to process and eventually like kick it out of the cell. Before we move on to the bacterial cell components, the attendance keyword of the week is flower, like the thing that you smell, the beautiful plant, Flower, flower. Flower is the attendance keyword of the week. Flower, the thing with petals, the plant. There is the attendance keyword of the week. Now, bacterial cell components are going to be similar, but have a few add-ons and a few subtractions. So the first major difference is going to be, while it does have a phospholipid bilayer, it has a cell wall. It's a wall on top of this bacterial membrane, of this phospholipid structure. Now, the purpose of the wall is to maintain the structure of the cell. It encapsulates the cell, it protects it from the external environment, and it helps it against pressure, so that way it's able to maintain its own pressure. It's like the multi-layers of an airplane, to where it's able to deal with all that pressure and it feels the same inside no matter what. That'd be the same thing for the bacteria. That cell wall is a double layer, so that way it protects itself against all of the external environment. Now, there's two major types of cell walls. There's gram-positive cell walls and gram-negative cell walls. And all of these are going to entirely depend on how much peptoglycan is actually going to be in that cell wall. The positive cell wall is going to be mostly peptoglycan, whereas a gram-negative cell wall is going to be some peptoglycan and then a lot of other structures as well as plasma. Here's a visual example of what that looks like. Here you can actually see, so take a note at the bottom on this gram-positive cell wall, you actually have the phospholipid bilayer down there. Cool, okay. Then there's gonna be this periplasmic space, and then that's like a glue or connective tissue effectively that would adhere the, well, it's not a tissue. Sorry, it is a glue effectively that is going to bind the phospholipid bilayer directly to, this is the most external area out here, outside of the cell wall. That's going to be all just extracellular space. This would be the inside of the cell down here. Now, this one has the peptoglycan wall. Look at all that. Look at those layers of that wall. And it's bound together with different acids that are going to bind these peptidoglycans together. So that's a gram-positive cell wall. Antibiotics can annihilate these things, no problem. Gram-negative cell wall, this is a little bit different. So again, we have the phospholipid bilayer, we have the periplasmic space with some peptidoglycans in here. And then we actually are going to have these different lipopolysaccharides. So it's a more complex structure on the outside. These are going to be more resistant to antibiotics because of their complex outer cell wall. So the antibiotics can't break down that acid and peptidoglycan chain as easily. Now, other components to kind of talk about are going to be, obviously there's the plasma membrane. This is the layer of phospholipids, semi-permeable, same thing as a eukaryote cell. We have the cytoplasm. The cytoplasm is going to be the gel-like substance inside of the actual phospholipid bilayer where all of the cell components will lie. Now, it does have a nucleoid. This is not a nucleus because there's no membrane around it. That's the difference. The nucleoid is simply the area where genetic material is going to free float in the cell. These are going to be the chromosomes containing the DNA. And you can see that here. Looks like a bundle of noodles or something. Few other structures we can note here are going to be things like, and I'll mention them and then talk about them, are going to be ribosomes that we can see here, the little dots. We have plasmid, little structure there. And then let me see if we can find it here. The inclusion bodies are going to be different storage sites that aren't pictured here, but we'll talk about them. So we have the ribosomes. Ribosomes are going to translate mRNA, that's a messenger RNA, into proteins and freestanding they are freestanding in the cytoplasm. So all these do, they act as kind of little programmers, little coders is what they do. They make more DNA. You have plasmids. These are going to be extra chromosomal circular DNA models, and they can code for survival enhancing genes. So these are specialized. The ribosomes themselves are going to be a constant that helps it just kind of deal with helps the bacteria do its general function, whereas the plasmids are going to be they're going to help it deal with different external structures. Like if an antibiotic comes in, this is how the bacteria learns how to fight it off. This is what allows it to learn and develop, are the plasmids. And finally, we have inclusion bodies. Inclusion bodies in there, imagine these are essentially like the cell's energy stores. That's all they are. They're an energy store for the cell so that that way it has energy to be able to move and go through its cellular processes. Now, that's the basics of bacterial cell components. Let's talk about the lab simulation. The lab simulation is pretty simple. You guys are going to identify and describe bacterial cells that you find in Antarctica. You're going to, a few reminders here. So pipette tips go in the red waste bin. Make sure, just as an extension from the pipetting lab, that you're always making sure you use the pipette once and throw it away. The moment you go to some other fluid, you need to dispose, you need to change and get a new pipette tip. You're also going to have this cool hollow table to be able to actually view the bacteria on in a larger structure. So that's pretty neat stuff. Okay. And lastly, this is kind of administratively, what is due by this Saturday at 11:59 p.m. is going to be the bacterial structure labster simulation. So the labster simulation, the attendance form keyword, I already said it earlier. And this is also a general reminder. There's no quiz this week, but in 2 weeks, week 7 in Cougar courses will be the midterm. The midterm is going to cover all the information from week 1 to week 6. So, and it's going to be 40 questions in total. And that'll be the only thing you'll do there. But I'm just making you aware so that way you're making sure you get caught up on everything. You're doing all the lab stirs. You're going through the PowerPoints. You're making sure you get everything done because that is coming soon. Other than that, go ahead and have a great week. I will answer a question. Just let me exit out of here. Go ahead, Raul.
Will there be a study guide for the midterm or no?
No, there is no study guide. The study guide itself is going to be each of the PowerPoints that you've gone through. And I'll tell you this right now, it's the same overall questions that you've gone through on the quizzes thus far. It's just all of those. Okay.
Okay. No problem. Thank you.
Have a good night, everybody.